Today's topic for the podcast is why belly fat is bad for the brain. And for this podcast, it's my pleasure to chat with a friend of mine and a former lab member a while ago, Professor Alexis Stranahan. She's Associate Professor of Neuroscience and Regenerative Medicine at Medical College of Georgia at Augusta University. And her work over the last couple of decades, it's focused a lot on what happens to the brain in obesity and diabetes, and because understanding that is critical to trying to develop ways, maybe even beyond counteracting obesity or diabetes and ameliorating the adverse effects of obesity and diabetes on the brain, which there are. And so maybe, Alexis, why don't you, I guess, before we start talking about this, your background. So you came to me, you were working on your PhD at Princeton, and you're doing work there on neurogenesis and effects of kind of stress, and I think social stress or something like that on the hippocampus and the production of nerve cells. And then somehow you saw our work and got interested in kind of doing the bulk of your PhD work in my lab at the NIH. Is that the way it happened?
Yeah, but even before that, I put myself through undergrad working as a fitness instructor and personal trainer. And I saw how people's, not only their bodies, but their minds, their emotional regulation would change with regular exercise. So at Princeton, I was looking at the intersection of wheel running exercise and social stress in rats. And I came across your paper. It was actually a review article called "Take Away My Food and Let Me Run." And I had really wanted to learn some more broad methods for analyzing synaptic plasticity, electrophysiology, some more molecular stuff. And I was really grateful and very excited to join your lab for the second half of my PhD training. And I have always appreciated your generosity in letting me kind of spin off a little bit on the obesity and exercise angle of things.
So Alexis was maybe in the top 2 graduate students I ever had. And as a graduate student, she was performing at levels that most postdocs. She was very independent, go-getter, and really tackled techniques, some of which actually you kind of learned on the fly, I think, in the lab. But why don't you briefly summarize your main findings from your PhD work with me, and actually that you extended you're kind of extending some of that to what you're still doing.
Oh, definitely. Yeah. So in my PhD research, we looked at how glucocorticoids, which are stress responsive hormones produced by the adrenal gland, how they regulate responses to positive stress in the case of exercise, or negative stress in the context of social dysfunction, or metabolic stress in the context of obesity and diabetes. So there's a bidirectional relationship between glucocorticoids and insulin sensitivity. So people with Cushing's disease, which is characterized by chronic hyperproduction of cortisol in humans, they gain visceral fat, and they often develop some degree of insulin resistance. And when treated, that goes away. And while in your lab, I had the opportunity to start working with this leptin receptor mutant mouse model. And this is a spontaneous mutation. It was originally reported in 1964 at Jackson Labs. And the mice lack receptors lack functional receptors for the adipocyte cytokine leptin. And leptin is produced by fat cells, white fat cells, And under normal physiological conditions, your brain detects that leptin and tells you, okay, we've got enough energy stored up, so we can stop eating. But when that receptor becomes desensitized, as in human obesity, or when it develops a loss of function mutation, as in the DB mouse, there's just protracted hyperphagia, increased food intake, severe obesity, insulin resistance, and cognitive impairment, as it turns out. So the work I did in your lab involved taking the adrenal glands out of the mouse, adrenalectomy, and giving back a low physiological replacement dose of corticosterone, the primary adrenal steroid in rodents. And it was important that we gave it back because a complete absence of glucocorticoids also causes hippocampal dysfunction. So it's not some linear relationship where more is bad, too much is bad, and too little is bad. So we clamped it within that normal physiological range. And the DB mice, they lost some weight and they restored their cognition. So they exhibit recuperation of performance in the water maze and object recognition tasks, and restoration of adult neurogenesis, which is also glucocorticoid regulated.
And yeah, and then, so the way that the cortisol acts on cells and neurons is it gets into the cell, it's lipophilic, it's derived from cholesterol. So it gets across membranes pretty easy and gets into cells, and then it will bind to protein that are transcription factors that regulate gene expression, turning on of genes, or in some cases turning off. Maybe we can get into that one with BDNF. But turns out there's actually two different receptors for cortisol. One, the glucocorticoid receptor, GR, and one is MR. And, and we talked about this, I think, when you were in my lab, that I had one graduate student, Jaewon Lee, who did this study with Jim Herman. It was actually when I was at Kentucky. And there was this kind of paradoxical situation where we put, or other people put rats or mice on caloric restriction or even intermittent fasting, which we did a lot of every other day, food deprivation. They live longer, they're healthier in many different ways, improved insulin sensitivity, et cetera. But they have elevated stress hormones, glucocorticoid. And so we're wondering, well, maybe the way that the neurons are responding to the cortisol or corticosterone are different with bad stress and good stress. And so the bottom line is, with bad stress and chronic psychosocial stress, levels of MR, mineralocorticoid receptor, go down and GR stays up. But Jaewon found was the opposite with caloric restriction or intermittent fasting. And then you did one experiment in my lab that kind of was consistent with that. Can you talk about that? You used it's a hormone that selectively activates MR.
Yeah. So the mineralocorticoid receptor has a very high affinity for corticosterone or cortisol, so it's tonically occupied. It also has a high affinity for aldosterone, which also gets into the brain. And the glucocorticoid receptor, because it has a lower affinity for adrenal steroids and minimal affinity for aldosterone, only gets activated if cort is up due to stress or at certain points during the circadian cycle when cort goes up. And so what we did in these experiments was, in this case, we're using a rat model of insulin deficient diabetes. So these rats had been rendered diabetic by injecting streptozotocin, which damages the insulin producing beta cells of the pancreas, producing a rat that is insulin deficient, normal weight. They sometimes lose some weight, and they have high levels of corticosterone. So we took those animals and put corticosterone, aldosterone, or a couple of agonists and antagonists targeting the mineralocorticoid or glucocorticoid receptor. We blocked MR with spironolactone, and we blocked GR with the progesterone receptor antagonist RU486, which also blocks glucocorticoid receptors. And we observed that activating MR actually restored long-term potentiation in slices from those insulin-deficient rats. Yeah. So that's interesting because so much in the glucocorticoid receptor biology field and the glucocorticoid neurobiology field focuses on the GR, yeah, as opposed to the MR.
Yeah. And I guess it's a good time. We've in previous podcasts with various scientists that done like 70 podcasts now, we talked about BDNF here and there. And so I guess it's a good time to introduce BDNF and the effects of the cortisol glucocorticoid on BDNF. Right. So I can't remember who initially, was it Bruce McEwen? I guess probably, that had the first evidence that glucocorticoid decrease.
Yes. BDNF? So actually there was multiple lines of evidence that all came out in, I believe, like the early to mid 90s with glucocorticoids and BDNF. So there was one line of evidence suggesting that a single exposure to high dose glucocorticoids, which is supposed to be like one of those flashbulb memories that you have from a single acute stressful event, and you encode that memory forever. Well, similarly in rodents, if you give a single high dose of glucocorticoids or a single acute stressor, you actually get enhanced BDNF expression. And that is thought to be GR mediated. And then, but with chronic stress, chronically elevated glucocorticoids, when that single acute stressor turns into repeated stress, the pattern changes. And in fact, the bound glucocorticoid receptor actually suppresses BDNF expression, not by, binding to the promoter in the classical glucocorticoid response element transcriptional repression mechanism. The GR makes a homodimer, and that homodimer sequesters the AP1 transcription factor complex. And since the AP1 complex is a major transcriptional activator for BDNF expression, that is how GR with chronic stress can suppress BDNF expression.
Okay. All right. And okay, so let's switch now. We'll jump to Georgia. And so you've been doing a lot with essentially looking at differential effects of abdominal fat and subcutaneous fat on the brain in models of obesity or diabetes. And so go ahead and start talking about that.
Sure. As an independent investigator, we've been focusing most recently on the role of the immune system, because obviously glucocorticoids have a major impact on immunity, and so does obesity. So obesity, I mean, there's a lot of individual variability. Someone like you, Mark, can eat probably whatever they want and doesn't gain weight, whereas other people, they just look at a cookie and 10 pounds comes on. But sooner or later, the laws of thermodynamics kick in, and that extra energy by consumption has to be stored somewhere. And it tends to be stored in adults as white fat, which is found in the subcutaneous compartment and in the visceral compartment. Now, even in lean individuals, the visceral fat has more macrophages, it has higher ambient concentrations of cytokines, and that's because it's all interwoven and intermingled with the gut, with the gut organs and the gut microbiome, which is theoretically separated from the gut, but in reality, there's always a small amount of leakage and interaction, causing that visceral component to be more pro-inflammatory. So with prolonged caloric overconsumption, that white fat expands and the adipocytes themselves get stressed. So they get overloaded with lipid and they start to put out these paracrine markers of cellular stress, and those attract immune cells, macrophages, into the fat. And initially they try to help, help keep these stressed adipocytes limping along, but with continued overconsumption, the adipocytes start to die. And at that point, the adipose tissue immune cells start to produce pro-inflammatory cytokines. And those cytokines actually play a critical role in the progression of peripheral comorbidities like diabetes, insulin resistance, or in some cases, hypertension. So that process occurs in parallel with another process. When those lipid containing adipocytes get overloaded, the lipid has to go somewhere, and it ends up in non-adipose tissues like the liver and the muscle. And those invading lipid droplets get recognized as non-self particles in those tissues, which further propagates the cycle of peripheral inflammation. And what we were interested in is knowing how this process of adipose tissue inflammation contributes to the cognitive effects of obesity in mouse models. And we weren't just interested in, visceral fat bad. Everybody knows visceral fat is a major risk factor for cardiovascular and metabolic comorbidities. But, is the subcutaneous fat just a passive place to store energy away from the visceral compartment? Or is it an active player that opposes the pro-inflammatory effects of visceral fat? So that was basically what I've been doing for the past 10 years, visceral fat bad, subcutaneous fat good. We take fat from either visceral or subcutaneous fat from a donor mouse, and we transplant it into a recipient mouse to see how signals from that specific fat pad impact hippocampal function and cognition.
And so you do this, what's the technique? You do this like liposuction type approach? Are you doing it?
It's more of
are you taking the fat from an anesthetized animal, or how are you?
Yeah. So it's a lipectomy. So we're not actually using suction. We're cutting it out, and we trim it to a fixed size so that, we don't have one mouse getting a big chunk of fat, another mouse getting a small chunk. Everybody gets 300 milligrams transplanted. That's just standard. And then by doing this and by using donor mice that lack certain genes, we've used NLRP3 knockout donors. And that's an interesting situation because the NLRP3 is a core component of the inflammasome complex, that it's a cytosolic protein assembly that, when formed, propagates and sustains the pro-inflammatory effects of obesity and, in some cases, injury in other tissues.
So when the inflammasome's activated, then pro-inflammatory cytokines are produced?
Yes.
And those include interleukin-1 beta and tumor necrosis factor, 1 beta and TNF. And you had so you had a paper in Journal of Neuroscience in 2014, Arian et al., where you provided pretty strong evidence that interleukin-1 beta was playing an important role in adverse effects of diabetes. You were using these same leptin receptor mutant mice that you used in my lab. Can you talk about that? And then, so I'd like you to go over like a couple, like 3 papers for sure. This one first, then your Journal of Clinical Investigation paper on the inflammasome, and then more recently, Nature Communications paper on what are called beige fat cells, beige adipocytes. But okay, talk about the interleukin-1 beta study.
Sure. So the 2014 J Neuro paper started out as an exercise study, and we wanted to. We actually used the amount of voluntary wheel running exhibited by DB mice in the studies that I did in your lab at NIA. Initially DB mice, when they're 6 to 8 weeks old, they'll run, it's only about 3K a week, but they will run in a voluntary running wheel. But as they get older and gain weight, they stop running voluntarily as much. So when we wanted to do exercise in my lab here in Georgia, we decided to use treadmill training. And treadmill training can be very stressful to mice, but we made 2 modifications to the protocol to reduce the stress. First, we did not use a shock grid to motivate them to run. Instead, we used compressed air, which was delivered manually each time the mice went to the back of the belt. And the other thing we did, which really put a cramp on everybody's social lives for a few months, was to do our training during the first part of the lights out part of the day. So mice are nocturnal, and we did the treadmill training during the first 2 hours of lights out. And we maintained that, 3 kilometers per week distance that was exhibited by the 6 to 8 week old DB mice in the running wheel.
Yeah, that's very important. That's something that a lot of scientists who use mice and rats kind of lose track of, the fact that, everybody's coming into the lab during the daytime, which is actually when the rats and mice are normally sleeping. So we're asking them essentially to, when if you do the running in the day or whatever in the day, that you're asking them to essentially lose sleep. And so, yeah. But anyway, so yeah, I can see where. Did you do it over the winter months so that it got dark earlier and they didn't have to stay so late in the lab?
So we did 3 cohorts, and I believe one was carried out during, the winter months, one during the spring, and one during the summer. So we did three sets of animals, training 7 days a week for 3 months. But it's 6 animals at a time on the mouse treadmill. So
yeah. Okay.
So that in the DB mice, it improved their insulin sensitivity. It restored the synaptic density, or the density of dendritic spines, which are the primary sites of excitatory postsynaptic contact in the adult brain, in the hippocampus of the DB mice, and it reduced their adiposity, and specifically their visceral adiposity. So after that, we went in and did lipectomy. So we surgically removed visceral fat from the DB mice, and we transplanted it into the wild type recipients. So this is just fat transplantation or fat removal, no exercise, because we wanted to mimic the effects of exercise on the visceral fat pads. And what we saw is that when we took visceral fat out of the DB mice, they exhibited cognitive rescue. And when we transplanted visceral fat from that DB donor into the wild type recipient, those recipients developed cognitive impairment.
And from the animals that were not exercised.
Right. Now, some other groups have actually taken fat transplants. In that case, it was a subcutaneous fat transplant from an exercising mouse and put it into a sedentary mouse, and reported some degree of benefit in terms of whole body metabolism. And that's another interesting series of studies, but we didn't do those. And I would certainly like to pursue it if NIH is willing. But with respect to the...
Oh, yeah. So you're going to talk about IL-1, interleukin-1 beta? Yeah.
Yeah. So the final study we did in that 2014 paper was to put a recombinant interleukin-1 receptor antagonist. So with most of the interleukins, your body either has a soluble receptor that floats around and soaks up all the ligand, or it has a soluble endogenous antagonist that soaks up the ligand, because you can't have uncontrolled inflammation all over the place. So we put that antagonist directly into the hippocampus, and we observed cognitive rescue in the DB mice, and that was our link to interleukin-1 as a mechanism for the types of changes that we were seeing, the synaptic loss and the LTP deficits.
And as these interleukin receptor antagonists, binds the interleukin. Are they used in the clinic at all for anything?
Yeah. They're used in the treatment of rheumatoid arthritis and I
Ah,
believe Crohn's disease.
ah,
So it's a biologic
yeah,
biologic therapy.
yeah,
And even though mice don't really have an endogenous interleukin-1 receptor antagonist, the IL-1RA still binds to mouse IL-1
yeah,
beta quite effectively.
yeah.
And we when we put it into the brain, we measured the infusions to make sure everything got in.
Yeah.
Um,
That seems like something that could be tried in Alzheimer's disease. I don't know.
yeah,
Even irrespective of, it gets across the blood-brain barrier, right? Uh,
um,
oh, maybe not.
that is uncertain because the drugs that it's treating in clinical use are peripheral disorders.
Okay. All
Um,
right. So in your experiments, it's essentially sopping up the peripheral interleukin, or
Probably. The thing is, the reason I'm saying probably is because the DB mice exhibit increased blood-brain barrier permeability and they eventually progress to blood-brain barrier breakdown. So I can't really tell you based on those studies whether, it's efflux, influx, or, whatever. But we did put it directly into the brain in those experiments. So we do know it was acting
okay.
centrally.
All right. All right. All right. And then continuing on the inflammation aspect, your JCI, Journal of Clinical Investigation study, where you already mentioned this, you have these mice that where the genes encoding NLRP3 are knocked out,
Yeah, yeah.
and you
So
use those to start looking at the inflammasome in the white fat, the belly fat, visceral fat.
yeah, and the NLRP3 knockout mice were a whole body knockout. And at the time we were initiating these experiments, there was no promoter that would distinguish between white fat in the visceral compartment and white fat in the subcutaneous compartment. And the currently available promoters are still more of a graded differential than a binary. But still, we wanted to selectively manipulate visceral adipose tissue inflammation, and that whole body NLRP3 knockout gains weight similarly to wild type mice on high fat diet, and they develop similar degrees of visceral adiposity. The main difference between the two strains is that they don't develop adipose tissue inflammation and they don't progress to insulin resistance the way that a wild type mouse on high fat diet would. So we took visceral fat from either a wild type mouse on high fat diet or a NLRP3 knockout on high fat diet, transplanted it into a lean wild type recipient. And the wild type mice that got transplants from the obese wild type donor developed brain inflammation, microglial activation, and cognitive deficits. But if the transplant, same size transplant, came from an NLRP3 knockout, that effect did not happen. There was no deficit at all.
And then in the same study, you manipulated interleukin-1 signaling only in the the microglia in the brain, which is essentially the equivalent of macrophages in the periphery, right?
Yeah, we were using the CX3CR1 Cree driver, and that Cree driver relies on time to generate specificity. Initially, when you knock something out, in this case, we knocked out interleukin-1 receptor 1, initially it hits all of the monocytes, macrophages, both in the brain and outside of the brain. But if you wait 6 to 8 weeks, those peripheral immune cells turn over, and the recombination is now restricted to microglia and other brain adjacent macrophage populations. So by doing that, we were able to narrow it down and say that, okay, visceral adipose NLRP3 is increasing local and circulating levels of IL-1, so that IL-1 is getting into the brain and it's acting on microglia to impair synaptic plasticity and cognition.
Yeah. really interesting. Are there any, okay, talk about then the beige beige adipocytes. Beige, you have white. Well, I guess talk about brown fat too first. Because mice, we when it gets cold, we shiver, right? And to generate heat. But mice have a lot of what's called brown fat, that the fat cells themselves, when it gets cold, they produce heat.
Yeah, they act more like muscle cells. They engage in thermogenesis. They induce certain mitochondrial uncoupling proteins. Yeah. And the thermal signature of brown fat can actually be read out in a mouse with a thermal camera.
Yeah.
And also, adult humans were initially thought to not retain any brown fat. It was thought to disappear during development. And then somebody did a PET study in Norwegian sheep herders that were always out in the cold. And they found the first thermal signature of brown fat in adults. And now with newer imaging technologies, they've found it in other individuals who aren't out in the cold herding sheep all day. But subcutaneous.
So these uncoupling proteins, so in the brown fat, they're packed with mitochondria. They have a lot more mitochondria than a typical fat cell. And the mitochondria have high levels of what's called uncoupling proteins. It's a protein in the inner membrane of the mitochondria that it uncouples oxidative phosphorylation from ATP production. So anyway, when it gets cold, these cells, instead of producing ATP so much, they're producing heat. And we actually, you didn't overlap any with Stephen Chan, right?
No.
No, he did some work with neuronal uncoupling proteins and found they were, it turns out you take cultured neurons, like I used to culture all the time in the lab, and you put them in the refrigerator and levels of the UCP4, it's a different uncoupling protein, but it does the same thing, goes up. And then we did some genetic manipulations of UCP4, and it has neuroprotective functions under kind of stressful conditions. Then we did experiments with old drug. Most people use maybe even in their first year undergraduate chemistry lab, if they have a lab, 2,4-dinitrophenol, or DNP. It's a chemical that uncouples oxidative phosphorylation. And a while ago, long time ago, people used to take this to lose weight. And they did. They lost a lot of weight. And then a few of them died because they said, oh,
Phenol.
Yeah, but essentially they essentially said, oh, if I take more, I'll lose weight faster. But then we found in low doses it's neuroprotective, and there's this company that we did some experiments with that's actually trying to develop DNP for a number of different neurological disorders because it's very neuroprotective. So the idea is it's inducing a mild stress on the cell by uncoupling oxidative, and then you get the cells respond adaptively by BDNF is upregulated, and certain other neuroprotective pathways. But anyway, so that's kind of interesting. So, okay, so brown fat, white fat, what's beige? It's between? Does it have more? Yeah, go ahead.
So the subcutaneous compartment, the white fat, the white adipocytes the pre-adipocytes can differentiate into white adipocytes that have similar physiological characteristics to visceral fat, or these beige adipocytes that behave more like classical brown fat. They induce the uncoupling proteins. Their structure is different. They have more mitochondria. They produce heat. And they actually stimulate a local anti-inflammatory kind of positive feedback loop, such that the immune cells in subcutaneous fat respond to signals associated with beiging, produced by both the beige adipocytes and by the sympathetic nerves that innervate subcutaneous fat. And that positive feedback loop kind of maintains and sustains the anti-inflammatory environment in subcutaneous fat. And loss of those characteristics, Bruce Spiegelman and Paul Cohen had shown that loss of those characteristics in subcutaneous fat, loss of beiging specifically, converts the subcutaneous fat environment into a pro-inflammatory microenvironment similar to the visceral compartment.
And is there, in people with obesity or type 2 diabetes, is there a reduction in the amount of subcutaneous beige fat?
People have used, actually Spiegelman's lab has used the patient tissue to demonstrate that with obesity, there is some change both in the local immune homeostasis and in the capacity for beiging in obese subcutaneous fat.
And then both exercise and caloric restriction will increase beige, amount of subcutaneous beige?
Yes, and actually extreme caloric restriction in the case of in rodent models of cancer cachexia can even stimulate beiging in visceral fat where it usually doesn't happen.
And then, okay, so talk about the Nature Communications paper. What did you do?
Well, we wanted to know whether is subcutaneous fat an active player or is it a passive place to store lipid away from this pro-inflammatory bad visceral compartment. So we took these mice that were generated in Bruce Spiegelman's lab. They lack the transcription factor called PRDM16. PRDM16 is necessary for beige fat differentiation in subcutaneous fat. And it's important to note that these mice express, they had a PRDM16 knockout under the adiponectin promoter, and that promoter doesn't get turned on until like early postnatal life. So the classical brown fat in this model forms normally. Spiegelman's lab reported it and we replicated it. So what they really lack is the capacity for beiging in adolescence and adulthood for a mouse. So we took these beige fat knockout mice and we put them on high fat diet and we looked at their cognition and we looked at a temporal window, which is 1 month high fat diet. Under those conditions, your wild type mice on high fat diet have not yet developed behavioral deficits, but the beige fat knockout mice did, suggesting that the loss. Yeah.
Did you do put glucose or fructose in their drinking water, or is it just high fat?
It was just the high fat chow, yeah, because you know, I did at one point write a grant to look at the role of high fat diet versus high fat diet with sugary drinks versus just sugary drinks. But I guess those types of parametric studies, or maybe I just didn't write it compellingly enough. So,
well, I mean, a lot of that sugar and fructose. There is evidence that fructose in particular promotes an increase in visceral fat, right?
Yes.
Yeah. So yeah, there's still a lot of people taking in a lot of fructose and glucose. So what about the translational aspect of this, right? Because transplantation, would it be practical? Of course, liposuction and bariatric surgery are for people who are like way obese are still used, but you think there's any potential applications for fat transplantation?
So people have plastic surgeons already use what's called homotransplantation for reconstructive surgery after breast cancer or severe accidents. So that involves relocating a patient's own subcutaneous fat to a different area. But what's really most relevant to this research is the negative findings arising from surgical, what they call omentectomy. Oftentimes, if a patient is undergoing some form of intragastric surgical procedure, if that patient happens to be obese, the surgeon will remove some fat and occasionally quite a bit of fat in order to be able to have access to the organ that they are targeting in their surgery. And it should be noted that multiple case reports, I mean, this hasn't been done in a widespread study, it's just, but all the case reports that I have read are consistently reporting that even removing, kilograms of omental fat as part of a surgery does not improve the patient's insulin sensitivity, at least during the temporal windows they look at. And
So
that's
by
because
omental, you mean the subcutaneous fat in the gut, in the belly, or what do you mean by omentum?
Oh, so I mean, like the visceral fat that is all interwoven around the intestines.
Okay.
So in mice, most of the visceral fat is found in the perigonadal fat pads. So it all kind of gloms together like a pad of butter. But in humans, of course, we have perigonadal fat, but we also have a more distributed network of
Okay.
white adipocytes in with the organs. So those negative findings are quite interesting. And they suggest one or two things. Number one, we're not looking at the right time scale. Maybe it just requires looking sooner or later than those case reports did. Number two, there could be compensation, because regardless of whether you take fat out of the visceral or subcutaneous compartment, and this has been shown in rodents and in humans, the remaining fat pads will compensate by growing. Yeah. So the translational implications there, I don't think are necessarily feasible or warrant pursuit. What's interesting in the subcutaneous fat story is that it turned out to be mediated by a set of T lymphocytes that are not in the brain, they're in the cerebrospinal fluid. And they're signaling from that cerebrospinal fluid compartment to microglia in the brain's parenchyma. So what we're seeing here is a peripheral cell type, T cells in this case, that is producing an anti-inflammatory factor, which we believe is probably IL-4. It's being produced locally in CSF because we measured it in CSF and in circulation. No change in circulation, increase in CSF after subcutaneous fat transplants. And T cells are uniquely amenable to being harvested from a patient, redirected in vitro, and programmed to attack certain things, and then put back into that same patient. So maybe that's a viable therapeutic approach for treating the neurobiological mechanisms underlying obesity itself and the cognitive and emotional sequelae of obesity.
So T cells produce interleukin-4, but are there other cells? Is there any evidence that neurons or other cells that might that IL-4 might end up in the cerebrospinal fluid?
Yeah, so there is some evidence from models of ischemic stroke and TBI that neurons, microglia, astrocytes have each been reported to induce IL-4 mRNA expression. We looked for it in obesity and we did not see it. So our current working hypothesis is that and this is consistent with the role of cerebrospinal fluid IL-4 in animal models of Alzheimer's disease. Our thought is that its production in CSF is acting within the brain, either via transport, volume transmission, or by acting on the epithelial cells lining the ventricles, and then those signal to cells in the brain itself.
Yeah. Because I think we talked about this before. We must have, or maybe not. So from an evolutionary perspective, okay, the macrophages, the innate immune system is pretty ancient. Maybe even before there were organisms that didn't really have nervous systems organized, but the humoral immune system clearly evolved after there was already nervous system. And we and others have found a lot of these quote unquote immune signaling pathways are operative neurons in the brain, toll-like receptors and many others, interleukins and so on. So I've always thought that maybe somehow these signaling pathways first evolved in nervous system, and then somehow when humoral system evolved, it kind of came from there.
Yeah, I mean, the nervous system and the immune system serve very similar functions, right? They have to be able to recognize stimuli, determine if those stimuli are harmful,
Yeah.
and then have some sort of effector mechanism to either pursue that stimuli or avoid it. And so in terms of functionality, it's basically two co-evolved systems, one in the brain, the other in the circulation and in specific tissues, that carry out similar functions. And I do actually think that humoral immunity, because I reviewed this recently for writing a literature review, and there is evidence that they co-evolved in tandem. I don't think that the humoral immune system predates or postdates the nervous system increase in nervous system functional complexity, because the certain Th2 cytokine loci, the cytokines that dampen inflammation as part of humoral immunity, those, I mean, that particular genomic locus is present going back to like planaria. So
Interesting. Now, one thing we haven't talked about yet is sex differences in fat. Women generally have a lot more subcutaneous fat than men. And so what are the implications of that for male-female differences in the impact of obesity or type 2 diabetes on the brain?
That is an excellent question. And we've recently started working in females. Initially, I was a little bit intimidated because I come from a neuroendocrine background. I know that so many things in the hippocampus and elsewhere in the brain fluctuate over the estrous cycle. So we spent probably a little over a year developing a mass spec-based assay to quantify sex steroids in mice. It's actually not as trivial as you might think, because the volume of sample is so low. Even with RIA, there have been some vendor-related changes in how the RIAs are made, and they've become less effective for mice specifically. So we did that by mass spec, and I had expected, given the literature on polycystic ovary syndrome, that females on high-fat diet would show some degree of interruption in their cycles, but that was not the case. We looked after, I believe it was 2 months, 6 months, or a year of high-fat diet in males and females, and we also had low-fat diet controls. And the females that were a year old, they both lost cycling at the same time, both the high-fat diet and low-fat diet females. We also looked at a number of different metabolic and neuroimmune parameters to see whether they varied over the estrous cycle. And at least under the conditions used in our study, they were fairly stable across the different stages. So after looking at that and determining that, we did a subcutaneous fat lipectomy in our females. And we did this actually before they started high-fat diet for a variety of reasons, trying to recapitulate the sex differences that are most prominent in lean mice by reducing that.
When you do that, I mean, taking out visceral fat seems relatively easy compared to subcutaneous. So you get, what did you say, 300 milligrams of fat?
Oh,
where do you take the subcutaneous? Because there's subcutaneous in a lot of different places.
right. So mice have the what are called the inguinal fat pads. Actually, the nomenclature is continually evolving. One of the things that's so excited about the new tissue clearing technologies is we are discovering zonation or anatomical subdivisions of tissues that were previously thought to be homogeneous. And subcutaneous fat is one of those tissues. So the dorsal lumbar component of the subcutaneous fat pad in mice goes basically up the back, as the name suggests, and then, but it's continuous with the inguinal component along the inner thigh. The component that is along the mouse's inner thigh has more adipocyte precursors, And if you leave a little bit behind, and by a little bit, I mean like 100 to 200 milligrams at that compartment, it will completely regenerate.
Mm.
So we removed the dorsal lumbar component, being careful not to disturb the inguinal lymph node and to avoid interfering or damaging the sympathetic innervation of that portion that is along the inside of the leg. And we did that before they started high-fat diet. So by the time we looked at them after 3 months on high-fat diet, the subcutaneous fat pad had completely grown back, and that was what we wanted to happen. We wanted to keep that a priori sex difference that would be found in an intact lean and an intact lean male and female mice. But what we observed was that there was no effect of subcutaneous fat removal on the time frame for development of insulin resistance in these studies. There was no change in sex steroids and no change in serum leptin levels. However, the females, which were originally refractory to obesity-induced microglial activation, so they were completely protected under intact conditions. But when you remove most of their subcutaneous fat before they start on high-fat diet, they develop microglial activation in the brain the same way that males do.
Huh. Interesting. And is there a role for, do you know what the signals are there from the periphery to the brain? Is it still interleukin-1 beta, or is...
We're looking at that, and I'm kind of hoping that NIH will renew my grant and we can continue to look at that. We do have some hypotheses. I mean, the whole female immune system is completely different than the males because the mammalian female immune system has to be able to cloak the fetus. So there needs to be a mechanism to shield the fetus from the mom's immune system so that she can carry to term. As a result, females have a much stronger anti-inflammatory Th2 bias, and that both contributes to females tend to have fewer side effects after the COVID vaccine, and that's thought to be Th2 mediated. Then again, females also develop autoimmune disorders at higher rates than males, which is the double-edged sword of having greater Th2 immunity. So we do think that it's that system that is mediating these effects.
Yeah, interesting. And so you mentioned, so I'm a grandfather now.
Oh, congratulations!
Yeah, just a little over 3 months ago. So Elliot's wife, whose name is also Alexis, her last name was Aduba. Her father was from Nigeria. And they had a baby girl, and everything seems to be good.
Oh,
yeah,
they're so cute that age.
And they're living close to us, so that's good. Let's see. I think we've covered a lot of ground here in a relatively short time. Did you want to talk any more? You briefly mentioned the blood-brain barrier when I asked about the interleukin-1 receptor antagonist, whether it gets into the brain. You have been doing some work on blood-brain barrier. You want to talk a little bit about that? And I guess, what is the blood-brain barrier first? And then what have you been looking at?
Well, the blood-brain barrier has different components. There's a structural component, which is the tight junctions that are formed between adjacent endothelial cells, and those really restrict paracellular permeability. So only factors that have specific transport mechanisms or lipophilic factors or very small aqueous soluble agents can actually get across without a transporter. So there's the structural component. And then there's an enzymatic component. So the surface of the endothelial cells will express certain enzymes that degrade certain factors or that suppress immune cells from sticking to the inside of the vessel. And then, of course, there's the parenchymal side of things. So you have the astrocyte end feet, which encircle the brain's blood vessels. And you also, depending on which type of vessel and where you are in the brain, there's also an overlying layer of vascular smooth muscle cells. And this is all critically important for brain health because the brain is an energy hog. I mean, it's a Cadillac Escalade with like all the trimmings and, you know, a 2-ton trailer pulling behind it. So you've got to have all this glucose and all this oxygen. Probably 70% of glucose and oxygen that you take in goes to the brain, and there's no local storage. I mean, astrocytes have some level of glycogen storage capacity, but unlike your peripheral tissues, you can't store energy substrates in the tissues. So any interruption can have really bad effects. And the astrocytes and endothelial cells cooperate to route that blood containing your glucose and oxygen to areas where there's high levels of activity in a process called neurovascular coupling. And I mean, obesity is known to have very widespread cardiovascular effects, most of them negative. And it's kind of not that surprising that these things are happening even more prominently in the brain. But the question is, we need to understand kind of how these things stage themselves. Is it what I think is going on is an initial increase in permeability that precedes the development of systemic insulin resistance, followed by outright loss of structural integrity after the onset of insulin resistance. And the reason that's translationally important to know, because obesity has been shown to induce blood-brain barrier breakdown in humans. They did a spinal fluid IgG measurement, and normally it's not supposed to be in spinal fluid, and it was elevated in obese individuals. So if we understand how this system degrades over time with obesity, then we can consider the best ways to titrate drugs that are not thought to get across the blood-brain barrier, because their CNS exposure and bioavailability is going to be higher in an individual with obesity and even higher in an individual with insulin resistance.
Yeah.
And that's kind of where we're going with that.
Okay. But have you looked at blood-brain barrier permeability in your different models where you're transplanting fat and so on?
We did it after visceral fat transplantation. It was one of the bazillion supplementary figures in that JCI paper, Journal of Clinical Investigation paper. And what we observed was that visceral fat transplants from a wild type mouse on high fat diet do not change blood-brain barrier permeability in the recipient, at least not in the time frame that we were looking at, basically.
So in that instance, there maybe not a blood-brain barrier leakage contributing to the adverse effects on brain plasticity, right?
Yeah, at least not at that time frame. But what we did see is a change in IL-1 beta transport. I mean, William Banks has done so much work over the course of his career. I've never met him, but he's like one of my heroes. He's the transporter guru, and characterized all these different transporters, including transporters that allow the penetration of, you know, picomolar concentrations of IL-1 beta to get it into the brain from circulation. And so what we observed is that range of transport is actually increased in the visceral fat transplant recipients, such that peripheral IL-1 beta can now gain greater access via these transporters into the CNS proper.
In our studies with intermittent fasting, of course, during fasting, eventually ketones will go up, right? You switched from using glucose to fats, fatty acids, and ketones, and there's ketone transporters, monocarboxylic acid transporters, MCTs, that move ketones, you know, into the blood, out of the blood, across the vascular endothelial cells and into the brain, and then maybe a different MCT transporting into neurons. It'd be interesting to look at those MCTs in some of your models and see if there's any effect on ketone transporter into the brain.
I want to say that Susan Minucci has done a lot with the ketone transporters, the MCTs. mostly in ischemia, but I think she has at least one reference in her very long set of PubMed pages that works in a model of obesity. And I've always thought that that's a really cool model. And the concept of energy substrate flexibility is actually a hot topic in pancreatic islet cell biology. So the islets need to be able to switch from using glucose to ketones and just different energy substrates. And I think that that concept would be really fascinating to apply to the brain, and understand exactly how that switch works. Because the developing brain, early postnatally, uses MCTs exclusively. I mean, breast milk is the natural ketogenic diet, and that neurons don't turn on the glucose transporters until the baby is weaned.
Yeah. And then the other end of life, so it looks like with mild cognitive impairment and Alzheimer's that, well, it's well known that glucose utilization is reduced. There's impaired ability of neurons to take up glucose, and we had some ideas on mechanisms for that involving membrane-associated oxidative stress and inhibition of glucose transporter. But apparently the neurons they're still there and alive, can still use ketones even though they have problems using glucose. So there's a number of clinical trials ongoing. There's actually already some published with medium chain triglyceride precursors of ketones, but there's now studies ongoing, including Dimitrios Kapagiannis, who's a neurologist, with me and patients with early Alzheimer's using a ketone ester. It's beta hydroxybutyrate coupled to this butene molecule, but it's essentially BHB, by beta hydroxybutyrate. And we found 10 years ago, we had a paper where we showed if we put this ketone ester in the diet, in the food, but in a mouse model of Alzheimer's disease, it had beneficial effects. So that's kind of interesting. And there's also radio tracers now for, of course, there's been for glucose for a long time, but Steve Conane up in Montreal developed radio-labeled acetoacetate, which is the other ketone. He does PET imaging in humans. But there's ways now you can look in the same individual at, relative glucose versus ketone utilization imaging. It's pretty cool. Yeah.
That is cool.
Yeah. Okay. So what is your most outstanding questions you'd like to answer, or question?
Oh, I think I'd like to know how inflammation, wrote broadly, is propagated from the peripheral tissues into the brain. Does it start at these circumventricular organs that lack a barrier and then spread out from there? Or does it start at focal regions where transport of circulating inflammatory cytokines is enhanced in some sort of pathology? And that's what we're trying to use these clear tissue technologies to get at. We want to understand how inflammation gets from the periphery into the central nervous system. And then once it's in the central nervous system, how does it get propagated from region to region? Does it spread along synaptically connected networks? Does it spread as a function of proximity? And we've been doing some things that I think were really exciting, trying to profile microglia morphology throughout the entire brain at micron scale using machine learning. So
and this is with the light sheet microscopy?
Yes.
We haven't touched on that yet at all, but it's essentially a way you can take a whole brain. Well, you don't have to do it in a whole brain, but it's in neuroscience. So I guess Carl who developed the method where you can essentially remove all the lipids?
The delipidation, yes. So Ali Ertuk was the one who reported in the 2011-2012 the IDISCO protocol, which was originally called DISCO, differential interference solvent something something. But it's a process whereby the brain gets dehydrated, lipid removal, and subsequent refractive index matching usually in a dibenzyl ether-based solution. And this process can be combined with immunohistochemistry. And since everything in the brain is so locally driven, now we can look from the olfactory bulb all the way back to the brainstem, and we can look algorithmically and computationally without tracing every single little brain region, which has always been a major rate limiting factor in these types of high throughput studies.
Yeah, previously you had to physically make sections, very small sections through the brain. With this method, you take the entire brain and prepare it as you described, and you can. So how does light sheet microscopy work then? This is how you're getting these and reconstructing the images?
Yes, so I mean, there are two main advantages of light sheet microscopy. Number one, it's faster than point scanning microscopy, like confocal or two photon would give you just a single point resolution. And that's, of course, a concern if you want to image at millimeter scale with micron resolution. And of course, it's less destructive because the tissue is being hit with a sheet of light instead of a point of light. There's far less photo bleaching. I've gone back
it's UV light?
So actually, no, you have to use wavelengths that are 560 and higher, because the lower wavelengths, your DAPI, your 488, are going to have much higher autofluorescence than your higher wavelengths.
Has anybody applied this to human tissue? Mouse or rat, their brains are pretty small. Has someone used the method, post-mortem on human tissue?
I imagine they have. It's been done with non-human primate tissue, both non-human primate brain, and actually I believe someone cleared an entire baby marmoset and imaged it with light sheet microscopy. So, and people have also taken cancer biopsies and cleared them and imaged them, which opens up another sample stream, because of course human tissue can be somewhat hard to come by in certain areas of research.
Yeah.
But the ability to deparaffinize existing biopsies or brain libraries or what have you and clear them and image them with light sheet is another whole frontier that can help us illuminate and maybe translate some of our findings from the rodents, non-human primates, and potentially into the human brain.
Do you have, I guess I didn't see it. Do you have a web page that I can put in the discussion? Do you have any examples of images from light sheet that you've done yet?
Sure. I can send movies. The department has a web page for our light sheet facility. I'm not sure how up to date it is.
And you're the director of the facility, right? Or you oversee the facility?
Yes.
Okay.
Yes, I'm in charge of the light sheet.
Okay, send me that, and then I'll also put links to the papers we talked about years and one review article, fairly recent one you wrote that I looked at in preparation for this. It's great to see you again, Alexis. It's been a while. Last time I saw you was when I retired from the NIA. It's already more than 4 years ago. Oh, so I've written 2 books since I retired. One on the science of intermittent fasting, but the other one, what you'll be interested in, is called Sculptor and Destroyer: Tales of Glutamate, the brain's most important neurotransmitter. So that comes out later this summer, and I'll send you a copy.
Yeah, that sounds really cool.
Yeah.
And if you have any medical students coming up through Hopkins that might want to come down and do some summer research.
I don't have a lab anymore, but maybe postdoc, because I teach 2 courses for the graduate neuroscience program. One on neurobiology of aging. They didn't have a course. These are electives for the graduate student. And the other is on brain bioenergetics. So yeah, I can, I'll try to remember that, direct them. But obviously there's a lot of good graduate students at Hopkins, and some may be interested in that, you know, what you're doing. So,
Yeah,
okay, say hi to Kyle.
I will. And congratulations again on becoming a grandpa.
Yeah. Okay. Bye, Alexis.
Bye.